So we are at this wonderful point in time when I have the pleasure to introduce our final speaker of the afternoon, Professor Carolyn or Dr. Carolyn Bertozzi, who was previously a Howard Hughes Medical Institute investigator before she moved to Stanford University in 2015. She was among the first faculty to join the Chemistry, Engineering, and Medicine for Human Health Interdisciplinary Institute, of which she is now the Baker Family Director. Her research group profiles changes in cell surface glycosylation associated with cancer, inflammation, and bacterial infection, and uses this information to develop new diagnostic and therapeutic approaches, in particular, most recently, in the area of immuno-oncology. Carolyn, thank you so much for joining us this afternoon. We are looking forward to your presentation titled Therapeutic Opportunities in Glycoscience. Welcome.
Thanks, Christine. It's great to be here. It's really exciting for me to get a chance to listen to all these early career investigators and to join a really amazing group of speakers and to kind of wrap it up for everyone. Can I share my screen now? Is that okay?
Perfect. Yes, please do. I love your background. That's
Thank
fantastic.
You. Yeah. Well, that's the theme of the talk. So what you're looking at here is the surface of a cell and all the glycoproteins and glycolipids that populate the cell surface. And that's an area of science that I've been interested in for the majority of my career. And today I'm going to tell you about some work that we've been doing in understanding how changes in cell surface glycosylation associated with cancer interact with the immune system, and some work that we've done to try to develop new immune therapies. So the title I sent you was kind of a generic title, but the more specific title is Targeting the Tumor Glycocalyx for Cancer Immune Therapy. And the story begins with an observation that dates back almost 70 years now, which is that if you take inventory of the structures of the glycans on healthy cells and cancerous tissue within the healthy tissue, there are some really striking differences. And back in the late 1950s, it was observed that one of the big changes that occurs in disease progression is that there's an overproduction of glycans that terminate with this particular sugar called sialic acid. So that in the lexicon of glycoscience is the pink diamond that's at the very tip of all of these branch structures. And this is the chemical structure. So the way I like to think of this is that all of our healthy cells have what I think of as like a well-manicured garden of sialoglycans. So those are glycans that terminate with sialic acid. But then if you look at cancerous tissue that emerges from those healthy cells, it's as if that well-manicured garden has turned into like a tropical rainforest. So there's more of these sialoglycans. Sometimes the structures have changed a bit, but there's just like a density of these structures. And this is true for almost every tumor type, both liquid tumors and solid tumors. So for more than half a century, people sat on this observation without having a good understanding as to why this would happen. And we started working on this now about, 12 or so years ago. And we were motivated to think about this because of the work that was going on in the field of cancer immunology back in the earlier part of the century. So we now know that the immune system plays a really important role in protecting us from cancer. And the way that works is that our immune cells patrol the body and they sample all of our cells one by one. And they do this through combinations of receptors and ligands that interact with each other at a synapse between the immune cell and the cancer cell. And the immune cell is basically looking for indicators of poor health, so signs that the cell has been damaged. And if the cell has been damaged, our immune cells will kill that cell and get rid of it, and that protects us from cancer most of the time. And when cancer prevails and when we develop a tumor, often that involves the ability of the cancer to undermine the immune system. So to show you a little bit more about what's going on at that synapse, and this is still quite oversimplified, but your immune cells have a collection of receptors that are activating receptors. So when those receptors engage ligands at a synapse with a cell, if the signal gets kicked off, then the immune cell will react. And depending on what type of immune cell it is, it will either kill that cancer cell directly or call for help so that other immune cells can come and help kill the cell. And the signals of damage that are presented by the cancer cell often take the form of presentation of neoantigens. And on the next slide, I'll show you a little bit more about what those might look like. Now, this is a bit of a treacherous situation because the cancer cell is one of your cells. It's just been damaged and needs to be eradicated. But because the immune cell can recognize one of your cells, there's always the risk that the immune cell might accidentally kill a healthy cell. And when that happens, you can develop an autoimmune disease. So the way that the immune cell manages that challenge is to have a complementary set of receptors that are recognizing signals of good health. And when those receptors get clustered at the synapse, they'll kick off a signaling cascade that actually inhibits the activity of the immune cell. So what's happening here is you have a group of receptors that are activating, a group of receptors that are inhibitory, and those two they're delivering signals to the immune cell, and the immune cell has to sort it out and integrate those signals and make a decision. Now, this was a very important development in the practice of oncology because people realized that cancer cells that are robust and can evade the immune system often do so by upregulating these signals here, like this orange shape, so that they can engage the inhibitory receptors. That's a smart move for a cancer cell, and cancer is a very evolutionary disease. So when cancers evolve this potential to drive the inhibitory signaling, they can live another day. And understanding that then led to a whole new class of immune therapies that block the inhibitory receptors. And the first two that were approved through this mechanism were the so-called T-cell checkpoint inhibitors. These are monoclonal antibody therapeutics that block either the receptor PD-1 on the T cell or CTLA-4, also on the T cell. And the biochemistry of how this works is rather interesting. So this slide shows even more detail at the specific synapse between the T cell and the cancer cell. So here I'm showing the activating receptor of the T cell, which is the T cell receptor or the TCR. And the T cell receptor will recognize on the cancer cell neoepitopes within short peptides that are presented by MHC molecules. And so remember that cancer always begins with damage to the DNA. And if that DNA is transcribed and then translated, there will be proteins or peptides that have mutations that reflect the DNA mutations. And those mutated peptides can be recognized as foreign by the T cell receptor. And this is a great way to activate the T cell to get rid of that damaged cell. But PD-1 and CTLA-4, the two checkpoint receptors, they do the opposite. They engage ligands that are on the cancer cell. So PD-L1 for PD-1, B7, and other family members for CTLA-4. And when these ligands bring the checkpoint receptors to the synapse, the inhibitory signal can dominate over the activating signal. And if you look at the cytosolic face of that synapse from the T cell's perspective, you can kind of understand the biochemistry of the activating and inhibitory signals. And it's basically a battle between kinases and phosphatases. So the T cell receptor has domains that are called ITAM domains. And these are domains that can recruit kinases and elicit a kinase cascade. And that's the activating signaling pathway. But the checkpoint receptors, PD-1 and CTLA-4, they do the opposite. They have domains that recruit phosphatases. In the case of PD-1, there are these two domains in the cytosolic region, the ITSM and the ITIM domain, and I won't bother you with what the acronyms mean, but they're docking sites for phosphatases called SHIP1 and SHIP2. So these phosphatases are taking off the phosphates that the signaling kinases would have put on, and it's that battle that ultimately leads to the decision-making in the T cell. So what the checkpoint inhibitors do, and so far they've all been monoclonal antibodies, is they either bind the receptor PD-1 or CTLA-4, or they bind the ligand, such as PD-L1, and they block the interaction of the inhibitory signaling receptors. And that allows the activating signal to dominate the response of the T cell. And you can see from all of these vials that there are some now very important drugs that have been approved for the treatment of cancer through this mechanism. The first of those was Keytruda, which is Merck's blockbuster drug. And Bristol Myers Squibb has a related antibody, Opdivo, and then Yervoy over on this side, the T cell. And then Roche was the first to have an approval for an anti-PD-L1 drug, and that's Tecentriq, and so on and so forth. And there's so many of these antibodies that are right now in the clinic that, I could give this talk a year from now and I'd have another, half a dozen vials on this screen. So this has probably been the most transformative breakthrough in oncology, at least in my lifetime. But everyone here probably realizes that this isn't a panacea. And even in cancer indications where patients might respond to these therapies, it's usually a minority of patients that have that benefit. But when a patient does benefit, they can have a cure to an otherwise fatal diagnosis like metastatic melanoma or stage 4 lung cancer. So why don't most patients actually respond to these checkpoint therapies? One theory is that the PD-1, PD-L1, and CTLA-4 axes, these checkpoint receptors on the T cell, they are not the only or even the predominant pathways for immune suppression in the tumor microenvironment, and that there are other mechanisms at play. And so back in around 2010, we started exploring other pathways that we thought might be important contributors to cancer immune suppression, with a focus on a family of receptors called the sialic acid binding immunoglobulin-like lectins. And that's a big mouthful, so we just abbreviate those the SIGLECs. SIGLECs are a family of receptors. There's about 14 of them in humans, and they are found on all classes of immune cells. So in this cartoon, the yellow line, you can picture that being the plasma membrane. And then outside the cell, each SIGLEC has a number of Ig domains capped with a domain that binds sialoglycans. So these are sialic acid, they're sugar binding receptors, which is why lectin is part of their acronym. And then if you look inside the cell in the cytosolic phase, this is what really struck us when we evaluated this family. Many members of the SIGLEC family have the two domains, the ITIM and the ITSM domain, that are also found in PD-1 and that have the ability to recruit those phosphatases to the synapse with the cancer cell. In fact, if you just take the cytosolic sequence of PD-1 and you do a bioinformatics analysis of the human proteome, you'll find that the closest relatives to PD-1 in the cytosol are these 4 members of the SIGLEC family, SIGLEC 7, 8, 9, and 10. They share a common signaling machinery. But whereas PD-1 is restricted to the T cell, you can find SIGLECs on all the different immune cells. So for example, SIGLEC 9 is quite prominent on myeloid cells, so macrophages, dendritic cells, neutrophils, activated T cells, and NK cells. These are all the kinds of cells that infiltrate tumors that we think of as being critical in the anti-tumor immune response. Same with SIGLEC 7. It's on NK cells, macrophages, and activated T cells, critical players in anti-tumor immunity. And so when you look at a cartoon like this one, and then you think back to how I started the talk, where there's 70 years of literature showing an overproduction of sialoglycans in cancer, you cannot help but analogize the PD-1 biology and the SIGLEC biology as potentially being relevant to cancer immune suppression. So just as the T cell has PD-1 that can engage PD-L1 on a cancer cell, leading to inhibition of the T cell, we speculated that maybe the reason so many cancers have an overpopulation of sialoglycans is because that phenotype allows them to engage the SIGLEC receptors on immune cells and suppress immune reactivity. And you can understand how by natural selection, that would be a phenotype that might arise over and over and over again in tumors from many different tissue types. So we set out to demonstrate this experimentally, and where we started was with the NK cell and also the macrophage, which express SIGLEC 7. That's one of the SIGLECs that looks a lot like PD-1. And these are cells that are able to react against a cancer cell through a process called antibody-dependent cell cytotoxicity, which immunologists would abbreviate ADCC. And this is an axis of immune reactivity that involves monoclonal or polyclonal antibodies. So as part of our natural anti-cancer response, our body generates antibodies against these neoantigens on the cancer cell. And if the cancer cell gets coated with these antibodies, that allows NK cells or macrophages to form a synapse through the activating receptor Fc gamma R3. And I haven't shown it here, but the activating signal that's delivered through this receptor is quite analogous to the T cell receptor. It involves many of the same kinase cascades. And so the question we asked was, if the cancer cell is overly sialylated, if it has that phenotype, is it capable of recruiting Siglecs to the synapse? So for example, Siglec-7, which is the preponderant Siglec on the NK cell. And if that happens, do the phosphatases recruited by the Siglec, Siglec-7, are they able to quench the signaling through Fc gamma R3 and protect the cancer cell against this ADCC reaction? And this is now published work, so you can read the details if you want. But the bottom line is that this is exactly what's going on. And it's not only the case for antibodies that are generated endogenously against the cancer, it's also a resistance mechanism against some very important cancer drugs. So for example, Herceptin, this is Roche's antibody against the breast cancer antigen HER2. So women who have a HER2 positive breast cancer, and I should point out, since this is a precision medicine symposium, Herceptin is one of the very earliest precision medicines, right? It's a medicine that a woman would get a prescription for if her tumor biopsy showed HER2 positivity. But even for women who have a lot of HER2 antigen expression on their breast cancers, about 50% of them actually don't respond to Herceptin. So there's a lot of clinical resistance, and really no one has known why. Well, it turns out if the cancer cell is overly sialylated and can engage Siglec-7, this quenches ADCC through Herceptin. So this is a resistance mechanism. And the same is true for another antibody called Rituxan, which is a leukemia drug. And so we think this is quite general. And it kind of got us thinking about, how would you deal with the situation where a woman is taking this medicine and not responding? And if you understood this to be a resistance pathway, could you develop an adjunct therapeutic that would promote reactivity against the primary drug? Anyway, so we published that article back in 2014. And since that time, many other papers have been published by many other important labs. And if you go through all of this literature over the pursuant 5 years, there's the picture that emerges, which is that the Siglecs, we think, are a legitimate and very important axis of immune suppression that one might want to target with a new generation of cancer immune therapies. And if you want all of this summarized in a nutshell, I thought Esther Landwees, who's a writer for CNE News, she wrote a really nice article about glycoscience and immunotherapy back in 2019. So that kind of brings us to the present day. And the challenge at hand now is understanding the biology as we do. Can we develop immune therapies that target the Siglec-sialoglycan axis of immune modulation? And of course, there's the playbook of the T cell checkpoint inhibitors, right, which have turned out to be blockbuster drugs. So, one could imagine making drugs that are analogous to Keytruda, where they're monoclonal antibodies that block the Siglec, right, the immune cell receptor, and prevent its engaging the sialoglycans on the cancer cell. The problem is that the Siglecs are a much more complex target than PD-1. So remember that there's 14 Siglecs in humans, whereas there's just one PD-1. And so you would have to know which Siglec, or more likely combinations of Siglecs, you would have to block to get a potent effect in activating the immune system. And since tumors are so individual, and even in one patient, each tumor has its own evolutionary history. So you could imagine that for any given tumor within a patient, there might be more emphasis on one or a combination of Siglecs than in another tumor, and it's going to vary from patient to patient. And so without knowing this, it kind of leaves you flying blind in developing a Siglec targeted therapeutic, which is why we decided to focus instead on targeting the ligands for the Siglecs that are on the cancer cell. So, what each Siglec binds precisely within a tumor microenvironment is still a kind of an open book. And this is something we work on and other people work on too. But what we do know for sure is, regardless of the precise sialoglycans, glycoproteins, and glycolipids that are supporting Siglec binding, every Siglec minimally requires a sialic acid residue as part of its binding motif. And there's been some structural biology on the Siglecs, and I'm showing here crystal structures and NMR structures of human Siglecs that are bound to various sialoglycan ligands. But the important take-home message is that although each one is different and they have a different footprint and they're recognizing different sugars underlying the sialic acid, they all have a hotspot where the sialic acid binds. That's what this orange circle refers to. And if you take the sialic acids off of their ligands, they lose a large amount of their binding affinity. So this gave us the idea that if we could just develop a therapeutic that you could park selectively on a cancer cell and that would somehow mow the sialic acids off the cell, which is why we think of these therapeutics as like lawnmowers. This would be a Siglec agnostic approach to immune therapy because every Siglec would lose its ligand if we took the sialic acids off. So this is what we're trying to achieve. And how do you make a lawnmower that will do this? Well, what we did is we thought of antibody-targeted sialidases. So a sialidase is an enzyme that catalyzes the hydrolysis of the bond between the sialic acid and what lies beneath it. And these are enzymes that are well known in nature. They're found in many species from bacteria all the way to humans. And so we made a molecule in which we took the sialidase from a bacterium called Salmonella, and we fused it to a tumor-targeting antibody. And in this case, we used trastuzumab. And that is the active ingredient in Herceptin, right? So this is the clinically approved breast cancer drug. This was a molecule whose predecessor was designed and constructed by Han Shao, who was a former postdoc, and now he's on the faculty at Rice University. And then Melissa Gray, who's a grad student who just completed her thesis a few weeks ago, built a sort of second generation molecule, which is the one that I'm showing here. So how did we put this molecule together? We did this chemically. And this turned out to be quite convenient for us because, in the early stages of this project, we were trying different antibodies and different sialidases and kind of mixing and matching. And it was really nice to have a modular approach where we could take different enzymes and different antibodies and put them together in different combinations. And the bioorthogonal chemistries that we're kind of known for in my lab turned out to be really convenient for this purpose. So what Melissa did is she took trastuzumab and genetically engineered near the C-terminus of the heavy chain the presentation of an aldehyde group. And I won't go through the details of how we do this, but we have a method for this where we just genetically encode a little amino acid motif that gets converted to an aldehyde in situ within cell culture. We call it the aldehyde tag technology. This sets up the antibody for a bioorthogonal reaction with dimethylhydrazine indole. And this is a chemistry that we call this the Pictet-Spengler ligation. It's kind of a riff on a 100-year-old classic from the organic chemistry literature. And it results in the formation of a carbon-carbon bond between the indole and the aldehyde. So this allowed us to put in a little tetraethylene glycol spacer with an azide group on the end. And everyone in the audience will know what you can do with an azide group, right? You can do click chemistry. So in parallel, we took the enzyme and then put a little spacer group onto the enzyme by selective alkylation of a cysteine residue. And that installed a cyclooctine, which is the complementary functional group that will click with the azide. So having enzymes functionalized in this way and antibodies functionalized in this way allowed us to click them together and make different combinations. So I think probably the most important experiment of all was showing that this antibody enzyme conjugate could selectively take the sialic acids off of cells that express the antigen, right, the HER2 positive breast cancer cells, without indiscriminately taking the sialic acids off all the other cells. And remember that it's important that we have Siglecs engaging our sialic acids to some extent, because that protects us from autoimmune reactivity. And if all of a sudden the sialic acids were blown off of all of our healthy cells, that would make it all the much easier for immune cells to react against them. And we don't want that. So to test the selectivity of the antibody enzyme conjugate, which we call T-SIA for trastuzumab sialidase, what Melissa did is she mixed together in the same tube HER2 positive cells and HER2 negative cells, and then asked at different doses whether this conjugate would selectively take the sialic acids off the HER2 positive cells. And she analyzed these cells by two-color flow cytometry. So what you're showing here is this is before the experiment. You can see the two cell populations. In this quadrant, these are the HER2 positive breast cancer cells. And then these are the HER2 negatives. And on the y-axis, we're staining the cells with a lectin called SNA that binds sialic acid. So that y-axis is a measure of how much sialic acid is on these cells. And they start out quite comparable. What's shown here is what happens to these cells after treatment with different concentrations, increasing from left to right, of the antibody sialidase conjugate. And you can see at the lowest doses that Melissa tried, so down sub-nanomolar doses, she almost quantitatively stripped the sialic acids off the HER2 positive cells. So they've dropped from the upper right quadrant down to the lower right quadrant. And by contrast, at doses all the way up to 120 nanomolar, there's almost no loss of sialic acid from the HER2 negative cells. So this is a really nice therapeutic window in which we can anticipate a molecule like this will take the sialic acids off only those cells that express the antigen that the antibody can bind to. And the reagent looks like it can potentiate the process of ADCC in a cell culture experiment. So what Melissa did here is she mixed together breast cancer cells that are HER2 positive with fresh NK cells from blood donors, and then she added these different antibody reagents. And you're looking at a function of dose of the antibody reagents. The trastuzumab sialidase conjugate is the most potent at eliciting cell killing in this assay. Whereas in control reagents, like the naked antibody trastuzumab, you get killing, but not nearly as potently. And this is a really great control. So what Melissa did is she took the sialidase conjugate and made a point mutation in the active site that renders it catalytically inactive. But otherwise, it's physically identical to T-sia. And this catalytically dead point mutant has no better activity than the naked antibody alone. So this shows that when you take the sialic acids off these breast cancer cells, they become vulnerable to killing by the NK cell in this experiment. So with that, we went into an animal model. And towards this end, we joined forces with a collaborator at University Hospital in Basel in Switzerland. This is Dr. Heinz Laubli. He's an oncologist who treats patients with immune therapies. And his grad student, Michael Stanzak, had developed a syngeneic orthotopic breast cancer model for HER2 positive cancers. So this is a model in which the murine cell line EMT6 is genetically engineered to express human HER2. And then he inoculates the mammary fat pad with these cells. And so the breast tumors are actually growing in the breast tissue. And so what we did in this experiment is allowed these tumors to progress to a certain point. And then we treated these animals by tail vein injection, either with the vehicle, PBS, with the naked antibody, trastuzumab, or at 2 different doses with the trastuzumab sialidase conjugate. And you can see that trastuzumab alone is completely ineffective in this model. So this is a trastuzumab resistant breast cancer model, and it was kind of known for that. And so we were able to recapitulate that. But if there's a sialidase on the trastuzumab, now the tumors, have a very hard time engrafting. And in fact, they're infiltrated with immune cells. And what Michael showed is that if you deplete these animals of their immune cells, then the tumors grow just fine. So this is an immune therapy mechanism that is specific to the presence of the sialidase. So with that, we thought it was time to really pursue human translation full force. And toward that end, I formed a company called Palion Pharmaceuticals, which is located in Waltham, which is a suburb of Boston. And Palion scientists have done a really wonderful job converting what was our original tool molecule, the sialidase from Salmonella, chemically linked, to the monoclonal antibody. And they've improved upon that molecule in 2 important ways. One, they made it much smaller by simply replacing one of the FAB domains with the sialidase so that they can retain the mass of a normal monoclonal antibody. And also, they figured out how to make this fusion genetically rather than chemically, which lends itself better to large-scale manufacturing. And importantly, rather than using the bacterial sialidase from Salmonella that we had used, which would probably be immunogenic in humans, they have now switched over to one of the human sialidases called NU2. And this is a molecule that's now in the GMP manufacturing stage for the first clinical trial. And we expect to have the first human dose administered in the first quarter of 2022. So that's just a few months away. So that's exciting, and we'll see how our hypothesis bears out in humans. But meanwhile, we thought that we could make even better refined therapeutics if we had more granularity in understanding the nature of the biological ligands that are engaging those Siglecs in the tumor environment. We know they're sialylated, because that's pretty much a universal requirement of the Siglecs, but we suspected that there was a lot more to it than just the presence or absence of sialic acids. So we've started working on some more fundamental questions having to do with what are the actual identities of these ligands on tumor cells. And the person who kicked this off was a Canadian postdoc named Simon Wisnowski, who just started his own lab at University of British Columbia just last year, basically in the summer months. And when he was a postdoc in my lab, what Simon did is to take a CRISPR screening approach to try to figure out what are all the genes that are essential for the production of Siglec ligands. And from those genes, can we infer what their molecular identities might be? So he started with a chronic myelogenous leukemia cell line called K562 that had been transduced by our collaborator, Michael Basic, with an open reading frame wide single guide RNA knockdown library. So each of the cells in this pooled library has a different open reading frame that's been knocked down by CRISPR-I. Then we could do a flow cytometry sort of these cells using a probe that is a soluble version of Siglec receptors. And these are Siglec ectodomains fused to an Fc tail and chemically modified with a fluorophore. And these reagents, by the way, are all commercially available for every human Siglec and most of the murine Siglecs as well. So he took two of these different Siglec Fcs, sorted this CRISPR knockdown library, amplified the library of the low expressors, and then deep sequenced the guide RNAs to figure out the identity of the genes. And what you're showing here are the genes that were required for production of ligands that bind Siglec 7 and also Siglec 9, where Siglec 7 genes or ligand genes are on the x-axis and Siglec 9 ligand genes are on the y-axis. So first of all, everything on the diagonal, those are going to be genes that are important for the ligands of both Siglecs. And it was gratifying for us to see essentially all of the genes for sialic acid biosynthesis popped up in this diagonal, which is exactly what you would expect. We also saw genes that are known to play a role in Golgi trafficking, which is also to be expected because these ligands have to get delivered to the plasma membrane through the secretory pathway. What was interesting was that we found a number of genes in this screen that were essential for the ligands of Siglec 7, these are the pink genes, and not at all important for the ligands of Siglec 9, showing that on this cell type, these two Siglecs, which are actually quite closely related to each other evolutionarily, they are binding very different molecules, other than the fact that they both have sialic acids on them. And in particular, Siglec 7 had a very strong requirement for the gene encoding a molecule called CD43, whereas this molecule was unimportant for the binding of Siglec 9. So we thought we should do a deeper dive on CD43 on the cell line and find out why it was an important ligand for Siglec 7. So CD43, first of all, it's what we call a mucin glycoprotein. What that means is that it has long extended regions that are rich in serine and threonine that are decorated with glycans on those side chains of those residues. And the glycans are so densely packed together for such a long stretch of amino acids that it forces the molecule to kind of stand up rigid and rod-like from the plasma membrane. That's a hallmark of mucins. It turns out that CD43 had an historical relationship with cancer. So this was known to be a marker found upregulated on leukemias, and the K562 cell is a leukemia-derived cell line, and also in solid tumors. And there were clinical data showing that CD43 expression is actually a negative prognostic indicator of the outcome in different disease settings. So for example, here's a study from diffuse large B-cell lymphoma showing that the patients that had higher levels of CD43 expression had worse overall survival and worse progression-free survival. So, and there's stories like this floating around the literature, but, you know, as is the case for most of these observational studies, no one really had a mechanistic interpretation of what this might mean. So we did a little bit of biochemistry to try and understand the composition of CD43 on the K562 cell. And to make a long story short, what Simon did is he made a bunch of truncation mutants to kind of winnow down where the binding was occurring. And it was quite surprising that all of the binding activity between CD43 and Siglec-7 was mediated by a little cluster of glycosylated serine and threonine residues right near the N-terminus of this large mucin glycoprotein. So there's a whole lot of glycosylation on this glycoprotein, but all that matters is this one cluster right near the end. It's almost like a flag on a flagpole. And what was quite striking about this cluster is that every threonine and every serine was glycosylated with the exact same tetrasaccharide. And it's a tetrasaccharide that's known in glycobiology vernacular as disiallyl T. And so here's the chemical structure where this T, the T antigen as it's called, refers to this disaccharide in black, where there's a galactose residue linked to an N-acetylgalactosamine residue. And then there's two sialic acids, one on the 3' position of the galactose, the other on the 6th position of the N-acetylgalactosamine. But this particular glycoform of CD43 is a high affinity binder of Siglec-7. It's quite striking. And when we saw this, we realized that if one wanted to develop an immune therapy that selectively targeted Siglec-7 ligands, what you really want to do is go after a structure like this. You don't need to blow every sialic acid off of the cell surface. All you really need to do is disrupt this motif. And, you know, we're at the case that blowing off sialic acids proved to have too many autoimmune side effects, because that's a pretty drastic thing to do. Perhaps a safer immune therapy would be one that targeted a narrower range of sialylated structures, for example, mucins like CD43. So I had a group of students in the lab who thought about these mucins and the role that they were playing in Siglec-7 binding. And they wondered whether we could develop a therapeutic that was not so much a lawnmower, but maybe more like a chainsaw. So could there be a degrader that we design that parks on the cancer cell and just cuts off the mucins like a chainsaw? And this was the goal of Keyvan Pedram, who was a former grad student. Now he's a group leader at Janelia Farm, the HHMI lab. And 3 current students, Judy Schoen, Gabby Tender, and Angel Kuo. And so where can you find a chainsaw? It turns out, similar to the sialidase, in the natural microbial world, there are proteases that have been identified that selectively cleave the polypeptide backbone of mucins. And these proteases are often called mucinases, and they're quite prevalent in bacteria that reside in the human gut. So we have a microbiome, and the bacteria in our gut, they have to live with us in symbiosis, and they have to be able to navigate an environment that is very rich in mucus. And mucus, which we all know is our natural lubrication, has its physical properties of being viscous and gel-like because it is rich in mucin glycoproteins that are secreted from the gut epithelium. So these mucinases are the bacteria's sort of hatchet. You know, this is how they get around. They cut up mucins very specifically. So what Judy and Gabby did is they cloned a whole bunch of these mucinases from human gut bacteria and profiled exactly what they cut, where they cut on mucins, and whether there's a reliance on certain types of glycostructures. And we've published a lot of that work, so I won't go through the details, but there was one mucinase in particular that we thought might be a nice starting point to develop a human therapeutic that targets cancer mucins. And that was the E. coli mucinase, known as STCE. So we call this enzyme sticky in our lab, STCE, we call it sticky. And what Stacy Malacher did, she's a former postdoc in the lab, is she collected a bunch of human mucins, digested them with sticky, and then by mass spectrometry, she analyzed where the cut sites were. And this allowed her to build a motif that is recognized by sticky. And what sticky likes to see is this tripeptide sequence where there are 2 threonines, The first threonine must be glycosylated, although the types of glycans it will tolerate are quite variable. And the second threonine can be glycosylated, but does not need to be glycosylated. And the residue between them can be highly variable. So that's a motif that you can pretty much count on finding within any human mucin. And sure enough, Stacy showed that sticky will cleave most human mucins into little fragments. And so this is just a blot showing the digestion of the human mucins MUC16, podocalyxin, importantly CD43, which we identified as being a high affinity binder of Siglec-7, PSGL1, which is a p-selectin binder, a couple of other mucins, and by contrast, proteins like BSA, which have no glycosylation, or glycoproteins like fetuin, which have glycosylation, but it's not the mucin type of structure. These are totally ignored by sticky. It's highly selective for mucins, and it will cut them. So the next challenge was to try and repurpose sticky as a targeted mucinase to deforest the Siglec-7 ligands on a tumor cell. And paradoxically, what we had to do was to actually damage the enzyme so it was less active than wild type. That's because sticky is really active. It can strip the mucins off of cells at very low concentrations. And part of the reason it's so active is that it has two domains. This is a crystal structure solved by Natalie Stranodka's lab back in 2012. The C domain and the so-called INS domain, these are domains that allow sticky to anchor onto cell surfaces. And once it's there, its catalytic domain, which is this metalloprotease domain, can just go to town and digest the mucins that are in proximity. And we didn't want indiscriminate cleaving of mucins off of healthy cells. We want to target this to cancer cells. So toward that end, what Gabby did is she said, let's take off these domains. And furthermore, let's do some mutagenesis around the active site to try and make a variant of sticky, which is almost dead. So it's a dead sticky. And so she removed those domains. We found an active site mutation that was quite effective. It was the mutation of tryptophan 366 to an alanine residue. And the combination of these three changes created an enzyme that had activity, but only at very high doses, and had negligible activity at the doses where the wild type enzyme could strip mucins off of cells. Then the goal was to restore the activity on target cells by fusing that mutated sticky to a tumor targeting element. And in the sialidase project, we had used the intact antibody trastuzumab. That's a large molecule. And we thought for this next variation on that theme, we would drop the molecular weight down and use an anti-HER2 nanobody, which was in the public domain and had been published. So here's the reagent that Gabby made. It's the nanobody with a spacer set of amino acids to our mutated, compromised, sticky variant. And so this is how the reagent behaves. So what you're looking at here is the ability of this and other reagents to cleave CD43, which we quantitate by flow cytometry, off of K562 cells, or K562 cells that we have engineered to express human HER2. So the wild type K562 cell does not express HER2. And you can see that when you treat wild type K562 cells with sticky, the wild type enzyme, at very low doses of enzyme, there's a quantitative removal of CD43. So we're down at sub-nanomolar, picomolar levels of sticky will strip CD43 right off the surface of these cells. By contrast, our mutant sticky, which is the green circles, has no such activity until you get way out into the hundreds of nanomolar concentration range. So the therapeutic window, so to speak, between wild type sticky and the mutant sticky is like 5 logs. Okay, so that's a nice window to work with. And our nanobody fused mutant sticky, which is the red circles, is no better than just the mutant sticky because there's no HER2 to target to on these cells. But now if you drop down here, when the K562 cells express HER2, there's no difference in the sticky activity, there's no difference just in the mutant sticky activity, but the nanobody fused mutant sticky, the red circles, have now gone all the way down in potency to match the wild type sticky. So we've compensated for the mutations that we've made by parking the sticky on the HER2 positive cells with a high local concentration with regard to the substrates. And now we can strip off the mucins. And we're now in the process of, I think we're about 2 weeks into 2 different tumor models to see whether stripping off the mucins with these reagents in the HER2 positive breast cancer model has any benefit in terms of potentiating the immune system. So we're going to find out soon. But in any event, hopefully you've gotten the big picture here. I started this talk with a description of our prior work in understanding how tumor sialoglycans engage immune cell Siglec receptors and drive immune suppression. And then I told you about some approaches to immune therapy that leverage this information. So, we've targeted the sialic acids in the tumor microenvironment for digestion with sialidases. And that's a concept that we will soon be testing in the setting of human clinical trials. And then I wrapped up with a description of a discrete Siglec ligand pair that we discovered, which is CD43 in this disialyl-T glycoform together with Siglec-7. We think that that is a paradigm that will play out for all of the Siglecs. And we're now in the process of looking for the professional ligands of other Siglecs. And this is a really active area of research in Simon's new lab at UBC. And then finally, we've taken an interest in mucin degraders, not just for cancer immune therapy. It turns out there are a number of human diseases which you could classify as a mucinopathy, where it makes sense to kind of target mucins for degradation in this extracellular space. So with that, I'll thank my group. I already mentioned by name the people who are directly responsible for the work that I showed, but of course there's a supporting cast that's very important in any lab. So this is my opportunity to thank everybody. And also to thank the people that supported this work, most importantly, the National Cancer Institute and the Howard Hughes Medical Institute. And with that, thank you very much. And I really appreciate being included in the symposium.
Wonderful. Thank you so much, Carolyn. What a brilliant talk. And I know that we are running very late. Would it be okay to just ask 1 or 2 questions? We have many questions, but maybe we could just cover one or two. Is that okay with you?
Sure.
Okay, wonderful. So this is a question from a PhD candidate in glycochemistry at Virginia Tech, Yang Zhu. And they're asking, I noticed there is a glycerol structure in sialic acid glycan. Can we use enzymes such as glycerol dehydrogenase to make the sialic acid glycan unrecognizable?
So first of all, that's a brilliant question. I really appreciate that question. And I think that is a perfectly reasonable proposal. I don't know much about glycerol dehydrogenases from natural sources and whether they would tolerate having one side of the glycerol moiety functionalized. And for those people who in the audience who might not be chemists and have no idea what we're talking about, let me get a chemical structure here. Am I still screen sharing?
Yep, perfect.
Okay, so you see this bit here at the end, there's OH, OH, OH. That's a glycerol group, except now there's a sugar hanging off of it, right? So to the extent that glycerol dehydrogenases might either naturally or might be evolved or engineered to recognize a functionalized glycerol, that would be an interesting approach. I can tell you there's a chemical method that people use to chemically label sialic acids on cells. And it's to do a mild periodate oxidation of the cell surface. And there are conditions people have worked out that will only oxidize with periodate, this primary diol of the side chain of sialic acid, without oxidizing other sugars, which wouldn't have a primary diol, but would have instead secondary diols. And that chemistry leads to basically a chopping off of the side chain and introduction of an aldehyde, which then people will do chemistry on the aldehyde with a biotin reagent or a fluorophore, right? And it has been shown actually for some of the Siglecs, when you do that periodate oxidation, that you can compromise the Siglec binding affinity. It's not true for all the Siglecs, but it's true for some of them. And so based on that precedent, I would surmise that if you had an enzyme that could perform a similar reaction and do something to modify that side chain, that you could have effects on Siglec binding affinity. That would be an interesting pursuit. So thank you for the question. It's a really, really interesting question.
Wonderful. Thank you. So in the interest of time, we will wrap up now. Thank you so much, Carolyn, for
Thank you.
sharing your research with us today. It was an absolutely brilliant talk. We really appreciate your time. In terms of closing remarks, I thank our keynote speakers, early career researchers, PRiME fellows, and innovation showcase speakers. You've made this a very inspiring symposium. Thank you also to Christine, Militza, and Angela for the tremendous job they've done in organizing this very successful event for us all today. Finally, I'd like to thank our supporters again, as I did earlier, the provost and our deans in the 4 constituent faculties: pharmacy, arts and science, medicine, and engineering. PRiME would not exist without your support. And finally, a big thank you to our attendees and dedicated PRiME community. And I hope to see you each in person soon. So thank you, everyone. Have a great week. Take care. Bye-bye.